as Megan said, it's based on a paper that within the working group we have made in the last month. Just actually, in the very beginning of the appearance of the pandemic, we decide that probably it will be interesting to have some knowledge or to revise the knowledge that we have about the potential transmission of the COVID-19, of the SARS-CoV-2, to the Antarctic wildlife, and to make a risk assessment to be used by the different stakeholders that has interest in Antarctica. So what the paper has been now in a preprint surveyed, but has now been submitted to a journal, and hopefully will be accepted. We hope in the next weeks. So the coronavirus disease, as all of you know, it's a pandemic caused by the severe acute respiratory syndrome coronavirus 2, the SARS-CoV-2. And the virus has spread rapidly to most parts of the world, having caused over 1 million confirmed deaths as October, unfortunately, and posing grave concerns to all aspects of human life, of course, with significant social and economic impacts, as all of you know. So the SARS-CoV-2 is a novel coronavirus closely related to coronaviruses found in bats and pangolins, suggesting a zoonotic origin with the potential for cross-species transmission among animals. So here is an illustration about the relationship, the genetic relationship between the SARS-CoV-2 coronavirus with other coronavirus found in animal species, in bats and in pangolins. But actually, so far, there is no direct identification of the animal species where the SARS-CoV-2 has the origin. So this is the closely related based on genetical relationships. So the virus can be transmitted from animals to human. This is because it's a zoonosis, but it can also be transmitted from human to other species. There is data of transmission to human from human to cats and ferrets, dogs and other felix like a tiger. In the case of cats and ferrets, it's also confirmed the transmission among the individuals of the same species, but it's not so far identified if can be transmitted from ferrets or cats or dogs to human. What is also clear is that, for instance, there is not any transmission among dogs, among themselves. And other interesting information that we will see in the next slides is that there is no any evidence of transmission or infection in pigs. But the most important for this talk is in any of the bird species that has been tried make experimental infections or has been follow the possibility of transmission from human to this species of birds. So considering that there is potential transmission from human to some species of animals, there is a question that if there is any possibility of transmission from humans to Antarctic wildlife, and this is the reason that we have made this risk assessment of the emergence of SARS-CoV-2 in Antarctic fauna. So we have paid attention to several aspects to try to have the global information to finally make an assessment of the risks of transmission of this coronavirus to the Antarctic fauna. First of all is the way of transmission that all of you know. Obviously, is transmitted human to human by the saliva droplets and also by the aerosol sprays. And this is the reason that the higher risks of transmission of contagious is in indoor places comparing with outdoor, and regarding the situation in Antarctica, where the most of the activity of the activities or the stay of the researchers and tourists in Antarctica is carried out in closed space, in the stations, in research ships, in camps, and also in tourist cruise ships. So, considering that there are closed space, the probability of transmission in case that someone has infected by SARS-CoV-2 could be very high. The other interesting point is to know what happened with the stability and effectivity of the virus, of this coronavirus, in the Antarctic environment. So what we know is that the SARS-CoV-2 stability is increased in cold conditions, and experiments conducted on other coronaviruses have demonstrated the higher stability of coronaviruses at extreme low or high levels of humidity. So there are some places in Antarctica that combine these two aspects, cold and high level of humidity in the coast. And therefore, Antarctic continent, some of the places, could provide suitable environmental conditions for the presence and transmission of the virus. So the areas where this can be more likely, beside the environmental condition, is the places where most of the human activity is carried out. And this area are where the research activity are conducted, and also where the tourists make their activity with the visits of the different places. So, and these places are concentrated in the South Shetland Islands, in northern Antarctic Peninsula, and in Victoria Land. As you can see in this picture, this is Victoria Land, this is Antarctic Peninsula, and this is the South Shetland, where all this activity that can be related with the transmission of COVID-19 would have the higher risk. So the most important thing is the transmission, direct transmission to Antarctic wildlife. Considering that the virus can be there in Antarctica, is how is the risk to that human can transmit the viruses in case there will be someone infected to the Antarctic fauna. So the most obvious thing is that the researchers, the wildlife researchers that has to capture and handling the animal, the wildlife, for instance, penguins, seals, they are the main group with the higher risk of transmission. So if one of these researchers has the virus, so the probability of transmission to the animals could be very, very, very high. Other human groups that stay in Antarctica, there are other scientists that has not any direct contact with the wildlife, and the tourists. In that cases, as the Antarctic Treaty, following the Antarctic Treaty rules, people that has not any permission for working directly with the animal has to be far than 5 meters far from the animal. So in this case, there will be no any risk of transmission because this is outside. So the aerosols would be dispersed by the wind, and there is enough distance to avoid the direct saliva droplets that can go from the human to the animal. But in some instances, for the rules allow that if one person is stand back without any movement, and one animal go to the person can maintain a close distance with the animal. And in that case, the risk of transmission could be high, just in case this person could be infected. So this is one of the things that we recommend in our mitigation measures, that in this next season should not be allowed for tourists or researchers that has not permission to work directly with the animal to maintain a distance less than 5 meters. In the case of other scientists that has no direct contact with the animals, the situation as well with other people from station, technicians, etc., is that there can be a encounter with the animals, accidental encounter that in that case could be increase the risk, but so we will have to be aware of this and to avoid this accidental to reduce the risk of accidental encounter with the fauna. Another aspect, another factor that could introduce the virus in the Antarctica is in this case is natural. Could be from animal migration. Species like a squid that migrate to the north, to South America, and in some cases even to Canada, could be in contact with the virus in this during the pandemic, and could carry this virus to the Antarctica, and when they are in contact with other species, they could transmit the virus. So this is another potential way of transmission of the virus. In this case, obviously, it's not human to animal; it's animal to animal. But could be important, and we will have to be aware of this potential way of transmission. Another way of transmissions that involve humans is through the wastewater. That is known that the viruses have been detected in fecal water, in fecal samples. So this year there will be pay attention to the treatment of wastewater to avoid that any residual in the water residual can go to the environment and can carry the virus and increase the potential of transmission to the fauna. So what happened with the virus with the Antarctic wildlife? Although we haven't a very well knowledge about the in general of disease, and I think that along this workshop you will see that the information that we have about disease, parasites and pathogens is still very little, is scarce, in some cases the information is very old, and in some cases the information is restricted to some geographical locations. But what we know now is that the virus are, of course, are present in the wildlife in Antarctica. Sorry. And in these pictures that belongs to the paper we are commented, we see that there are several coronaviruses genera that has been found in different species in Antarctica, and also in another close species not in Antarctica but in the Arctic, and the other kind of viruses that are present in different species of penguins, seals, and other seabirds. So I think this is a very good occasion to increase our knowledge about the presence of virus in especially, but also on disease, following this pandemic, and considering the importance that this information has in the preservation of the wildlife in Antarctica. So going to how is the susceptibility of Antarctic wildlife to infection by SARS-CoV-2? What we know first is that we don't know nothing about this coronavirus in the Antarctic wildlife, because there is no any opportunity to make any analysis, to take any samples to know if this coronavirus can be already in the Antarctic fauna. It's very unlikely, but we don't know nothing about this. So the information that we have is coming from theoretical, from in silico analysis that by this author, Davos et al., that they made an approach, made comparison of the structural analysis of the AC2. This is the receptor that the coronavirus use to go into the cells and to infect the cells and to spread the infection into the body of the host. And what they have made is to compare this structural structure of this receptor in different animals, in several around 400 species. This is an example of how the results are shown in this paper. And considering the probability that the structure of the receptor can be fit with the structure of the virus, they make an assessment and they make about the risk of infection of the different species. So there is four species of Antarctic species that has been included in this study. But a summary of the results, they found that the Cetaceans are the group with the higher risk of infection because of the close relationship of the close how the close the receptor the receptor is in the host to the structure of the virus. So the Cetacean have the higher risk. The pinnipeds have a very low risk because it means that the virus, considering this structure, would be very, very difficult that can be infect this species. And in the case of seabirds, including penguins, there is also very, very low risk. As I said, there has been several species included in the study. Two of them are the Adderley penguins and the emperor penguins that show a very low risk of relationship between the structure of the receptor and the virus. And in the case of the Cetaceans, there are one species, the minke whale, that show a very high risk and is also present in Antarctica. What is also interesting to know is, as I commented, as I said before, is that there is no any evidence of birds can be infected, even through experimental infection, infected by the virus. So actually, the risk of the birds to be infected is really very, very low. So to summarize, we can conclude that the conditions in Antarctica could be favorable for SARS-CoV-2 stability. In silico analysis revealed that cetaceans have a higher susceptibility to the virus. Seals and birds seem to have a lower risk of infection, and wildlife researchers have the higher risk of transmission to Antarctic fauna. And tourists can be potential vectors for SARS-CoV-2 transmission to fauna. I forgot to mention, I go back. Okay, I forgot to mention here that these results based on the in silico analysis should be taken with caution, because one of the species of bat species that is thought that can harbor this kind of coronavirus in the in silico analysis also show a very low risk of infection, when we know that the coronavirus can be found in this species of bats. So it's probably that there is no just this ACE2 receptor that was involved in the infection of the virus to the cells, and can be another receptors involved, and so this is just the information that we have now, but we will have to take with caution this result and this risk. So in that case, we thought that we have to use the precautionary principle, and until having more information, we will take the situation as every species in Antarctica has a considerable risk to be infected by the SARS-CoV-2. So, as I said, this is the summary of the results, and this is a picture that try to illustrate how can be all the ways or the contacts between the tourists and researchers to wildlife, infection human by human, and potential transmission to the fauna by the sewage, or by direct contact by handling for direct work, or for accidental encounters, and also taking into account the potential transmission by migratory species. And I think this is all for my part. If you have any question, I will be happy to answer. So thanks for your attention.
Perfect. Thank you, Andreas. So as I said earlier, we have got the Q&A box. So if you have any questions, please pop them in there. And once we get through our next two talks, we'll open up for questions for that first session. So our next speaker is Michelle Power, who's going to talk about the workshop that was held in Sydney, looking at microbe-parasite interactions in Antarctic wildlife. So Michelle, when you're ready, if you'd like to share your screen.
Thanks, Megan. Welcome, everyone. And thanks, Andre, for that excellent talk. So I've been asked to talk about the Sydney workshop. And before I get into some of that, I'd just like to give a bit of background on the research that I've been doing that set us up for that workshop. And that's basically the dissemination of antimicrobial resistant determinants via Antarctic sewage. And before I start, I'd like to acknowledge the traditional Aboriginal custodians of the land that I am currently on, and that's the Gadigal people of the Eora Nation. And I'd also like to pay my respects to elders past, present and emerging, and particularly any Aboriginal or Torres Strait Islander people that might have joined the webinar today. Okay, can I just ask for people to mute microphones because I can hear some background noise? Thank you. All right, so I'd just like to give a bit of background, and this touches on some of the aspects that Andre has brought through in his talk, and that basically global change, movement of people, all sorts of activities are driving microbial traffic. And this sits in the One Health concept that basically wildlife, domestic animals and humans are all connected. So not only are we connected, the pathogens or parasites that we carry are also becoming more connected. And the current pandemic is a fantastic example of this connection. So things that people do drive this microbial traffic. So if we think about the three spheres here with domestic animals, wildlife and humans, we particularly impact wildlife through a whole range of activities, and including translocation, conservation practice, urbanization, travel, tourism. And the more we connect with wildlife, the more we connect with pathogens and parasites. And this is all outlined in Peter Daszak's paper presented in Science in 2000. And it sits basically, or represents the drivers of emerging infectious disease. Now, interestingly, we can think about antimicrobial resistance as a emerging pathogen or suite of pathogens or emerging disease issue. And also the emergence of antimicrobial resistance follows that One Health concept. And just to clarify, antimicrobial resistance is any microbe demonstrating any pathogen class, any taxonomic class of microbes demonstrating resistance to chemicals or medicines that we use to treat the diseases they cause. And when we talk about antibiotic resistance, we're specifically talking about the resistance of bacteria to antibiotics or those drugs that we use to control them. All right, so wildlife, as we know, on the urban fringe. Here's some examples in Australia that we have very close to our cities or smack bang in the middle of our cities. So bats, possums, kangaroos, and little penguins. And then some examples from some of the countries that might be where the participants are here today. So sea otters, opossums, raccoons, and squirrels. And all these species, you know, growing more and more, there's plenty more, these are just some examples that I grabbed, are growing more and more connected to people. And as I said, the pathogens we carry and setting up for transfer of those organisms that people have. And of course, there's also an urban aspect to Antarctica. And so we have all the research bases that are distributed throughout the continent and sub-Antarctic areas, and also the fast-growing tourism activities that are occurring down there. And obviously, when humans visit Antarctica, so do our microorganisms. And so back in, oh gosh, look, this was a dream I had since before my PhD that came into reality around 2008 when we received some funding to actually go and look at the transfer of microorganisms and particularly antibiotic resistant bacteria from humans and their dissemination into the Antarctic environment at Davos Station. So some of the questions that we were interested in asking, you know, what microorganisms are humans leaving behind? And we've just heard an interesting story from Andre and potential risks about what we might take in. And then also, what are the potential impacts of those microorganisms to Antarctic wildlife and the Antarctic ecosystem as a whole? So we were focusing our study on a genetic element that has been particularly important for the dissemination of antibiotic resistance with gram-negative bacterial pathogens, so Klebsiella, E. coli, Salmonella. It's a naturally occurring genetic element that has really been associated with bacterial species for many millions of years, and it has just, over the period of humans' massive use of antimicrobials, it's been placed under selective pressure and has now become a primary mechanism for encoding antimicrobial or antibiotic resistance and also disseminating it. So integrons are both a gene acquisition and expression system. And basically, I'll just talk through this little picture. Got a bacteria here, and this is the class 1 integron. The integron has a conserved region and the ability to insert genes. So we can see here, it's grabbed this green gene here, insert genes into what we would term an array, and then immediately express those or turn those on and gain the trait that that gene encodes. So we can see here this gene represents X antibiotic, okay? And so once this bacteria acquires the green gene, it now has resistance to that particular antibiotic. But the environment has an entire suite of resistance genes called the resistome that encodes resistance to diverse classes of antibiotics. And when bacteria are stressed from whatever reason, they will start sampling that environment and literally grabbing their genes or the genes from the environment, and then they can rapidly insert them and express them into this system if they're carrying the genetic determinant and end up with multi-drug resistance. And now the class 1 integron is widespread throughout humans around the world, but also the environment, wildlife and domestic animals, and an important disseminator of resistance. So not only can the class 1 integron transfer antibiotic resistance traits between or acquire from the environment, it can also transfer those resistance traits between species of the same individuals of the same bacterial species, but also to individuals of other bacterial species. And this is one of the reasons why it's been played such a major role in the emergence of antimicrobial resistance among those gram-negative bacteria or gut bacteria or certain species of gut bacteria. All right, so we were asking, is there evidence of these genetic traits that are associated with humans in the Antarctic environment? And this here is a picture from Casey Station, and basically you can see the pipeline here, the sewage outlet. It's constantly heated to allow or to ensure that you don't have ice blocks of sewage building up and then disposed of usually into marine waters nearby. Now, the Antarctic Treaty does have some guidelines on sewage disposal and waste disposal, and that it needs to be in a manner that doesn't adversely affect the local environment, and that reasonable steps are taken to discharge sewage or waste into the sea at a place where conditions exist for dilution and rapid dispersal. So at Davos Station, at that time, the sewage was macerated, so basically broken down in fine particles, but beyond that there were no further treatments, and it was being disposed of into the marine environment. You can see the aurora out here, the kind of, what do you call it, where, I've forgotten the name of where we get off boats when you get off a boat. It's just around the corner here anyway, not very far. And I always found this amusing that the plumbers who were there awaiting our arrival for sampling set up this little sampling depot of a 44-gallon drum when really we're only after about a litre or so. So that's kind of the situation at Davos. There's a number of, depending on how many people are at a site and where those sites are located, the guidelines vary. But there's basically a lot of untreated fecal material and microorganisms that actually do survive the harsh conditions in Antarctica that we're leaving behind. So we approached a microbial profiling aspect to look at human impacts. We were looking for the presence of resistance determinants that I've just mentioned. And we also were doing some additional characteristics where we were looking at evolutionary lineages of bacteria or strains, and also working out whether we were targeting E. coli, whether our isolates had traits that suggested that they were human-associated strains, or E. coli that we might expect to see in vertebrates in Antarctica. Just briefly, the methods: we were collecting all sorts of samples, sediment, core, water, feces in vertebrates and vertebrates. We were doing coliform counts on water or environmental samples, and then we were also isolating E. coli from our range of sample sources, and then doing molecular tests to look for those antibiotic resistance mechanisms and characterize the strains. We found that 12% of our E. coli isolates were positive for those class 1 integrons and carrying antimicrobial resistance genes. So in this table, you can see the numbers of E. coli, the sources of those E. coli, and then over here, these are the class 1 integron constructs, and basically you can see that there's diverse resistance genes. So here, AADs, DFRAs, well, 2 types, but they encode resistance to 2 drug classes, but there's diversity within those genes. So basically this shows that there's quite an array of different types of antibiotic resistance genes associated with this class 1 integron that are distributed throughout the Antarctic environment. So the big one, we cultured E. coli from Antarctic wildlife, but the E. coli that we looked at were not carrying class 1 integrons, so we didn't find any evidence of resistant E. coli in the samples that we had, but we did find E. coli that were representative of emerging clonal strains that are associated with humans, and these are the sequence types 131 and 95. These are known to be multidrug resistant outside of Antarctica, and ST195 is a pathogen of birds outside of Antarctica. And if you want to look at the results in the study in more detail, you can grab the paper. All right, so that was the background to pretty much my research in Antarctica and how I connected with this group. And it actually started flagging wider issues that, you know, we're seeing human-derived E. coli being surviving in the Antarctic environment. I think there's been a kind of idea that the cold temperatures, the extreme conditions that bacteria, perhaps from warmer areas and from us, will actually not survive those conditions. I think, you know, bacteria rule the world. So it's pretty clear they're surviving down there. There's an accumulation of human-associated E. coli in marine invertebrates, and these are resistant bacteria. And so now we have antimicrobial resistant E. coli associated with the food chain. And then the genetic elements are of significant for bacterial evolution, have also now been placed under huge human selection pressure, have been transported to Antarctica. And then this whole system to back then, the idea that it sets up a way, perhaps a rapid screening tool for across the continent to measure sewage pollution, but it also potential indicator of what humans are introducing into that environment or what microbes we're leaving behind. So this really led to the Antarctic and Microbial Impacts Workshop and a meeting of the Wildlife Health Group, and it was sponsored by Macquarie University, Genes to Geosciences, and SCAR also now. I think that was the conversion for the contribution from SCAR. So the workshop aimed to bring together a cross-disciplinary team to, look at some of the aspects of the introduction of organisms, both bacterial and parasites, to the Antarctic environment, and to also start considering how that the introduction of those organisms might impact the native symbiotic interactions in wildlife. So the specific goals were to generate a review on microbial threats arising from human impacts in Antarctica and the application of emerging molecular technologies, and consider multi-layered symbiotic interactions as tools for investigation. Further, to generate 2 reviews on disease risks to Antarctic invertebrate groups, and to think about a strategic plan and large-scale grant applications and collaborative research. So, what have been the outcomes? It actually resulted, when I was reflecting and looking back, I think it wasn't only this group's members who came, we had other people from Sydney and Australia join, and it's connected people, members of this group, who have continued to ask some really interesting questions. It facilitated and supported research between participants in other areas, and also other research areas. And also we had some people dropping in from different parts of the country, and some of the group members trekking out to see others, and it facilitated research outside or with non-participant workshop people. We identified knowledge gaps and outputs to address, sorry, I'm not sure why that line's dropped off. We identified knowledge gaps and outputs to address the issues that we identified, and we've had also identified gaps that still stand strong today and represent goals of the working group. And so that was to catalogue pathogens and parasites, to enable baseline data, and to also look at the application of emerging molecular technologies to understand Antarctic wildlife health and disease risks. And I think some of the aspects and outcomes of this workshop will be reflected in some of the further talks today. Okay, so I'd just like to thank you all, and if there's any questions, post them in the chat and we'll address them at the end, as Megan pointed out. Thanks, Megan.
Perfect. Thank you very much, Michelle. So our next speaker for this session is Thierry Boulinier, who's going to talk about our polar wildlife health workshop we held at the Polar 2018 conference in Davos.
Thank you, Megan. Hi to everybody. I'm just setting up the screen. So following the previous talks, I'll be giving a talk about a summary of the second meeting we had, and which happened during the Davos Polar 2018 meeting, so in Switzerland, where we focused on wildlife disease and addressed different issues. Particularity of this meeting, is that it considered both the Arctic and Antarctic and subantarctic areas. And so it was entitled Connecting Ecology, Health and Disease Issues in a Changing World in Polar Wildlife. It was sponsored by IASC, International Arctic Science Committee, Terrestrial Working Group, notably, and got also some more smaller sponsors. And it was organized by a broad group of people, with Susan Kutz from University of Calgary, Andres Barbosa from a museum Madrid, Megan, who is superbly organizing the webinar today, and myself. And basically, we wanted to make an overview of what was going on polar wildlife issues. And we focused on first on the issue of monitoring wildlife disease. Why monitoring wildlife disease? What to monitor when you want to monitor wildlife disease, and how to do it. And then we address a series of global issues, and as well as case studies, and had a good discussion about all these different topics. So following on some issues that had been raised by the previous meeting, and opening perspectives. The attendance was large, but not huge. Of notice, there was several postdocs and PhD students, some from Canada, Pratap Kaffel, Fabien Mavro, Amandine Gamble from France, notably, who contributed talks to the meeting, which was quite nice. And we dealt with very different types of parasites and pathogens in wildlife. I just put this slide full of seabird tick, Ixodes uriae, just to stress that this is one parasite that's found both in the north, in polar North Pole, polar areas in the north, as well as in the south, in seabird colonies. And so this kind of organism can be studied in very, very remote areas, at two extremities of the Earth, but it's facing the same kind of conditions in these environments. So part of the discussion we had, we raised questions about difference between what's going on on the North Pole in host-parasite interaction and in the southern polar areas, with a big difference, of course, in the landmass and in the occurrence of many terrestrial mammals, natural occurrence of terrestrial mammals in the north, which have host pathogen interactions, much less terrestrial mammals on islands in the south, but introduced mammals in some of the islands in the south that can be involved in epidemiological cycles. And so I'll just, I guess, go a bit fast, but through the different talks that were given, just to give you an idea of what kind of topics were addressed. So Susan Kust gave an overview of Arctic wildlife health issues, stressing the importance of changes in the environments and how this cascade down to host parasite interaction, host pathogen interaction. Andres Barbosa discussed how we should update our protocols, field protocols, to gather samples on these issues. So both in Antarctic and subantarctic field sites to get to be able to gather samples that will be useful to address issues at broader scales. Megan gave an update on the new technological tools that were available, notably genomic tools available for disease surveillance. And I gave a talk about an old tool that can be used, which is serology, the ability to detect antibodies against infectious agent in vertebrates, which is a relatively old approach, but which can still be very useful to detect infectious agent against which some individuals may have been exposed, and which basically relies on samples, blood samples, to look for these antibodies in the plasma or the serum of the wildlife that has been sampled. And finally, Emily Jenkins from Canada, she focused in the north, in the Arctic areas, on the issues of monitoring zoonosis in relation to people that are living in these areas and that may be hunting animals that can carry and transmit parasites and pathogens. So we had quite a complete overview of many different topics. And then we had a series of case studies on these issues. We had 2 talks involving ungulates in the north with a disease in muskox in Canadian Arctic and nematodes in Arctic ungulates, which stressed the importance of understanding the dynamics of the environment in relation to the occurrence and exposure to parasites and the effect of the parasites on these vertebrate hosts. Then we had a series of talks about seabirds, about dispersal, of the role of seabirds involved in dispersing infectious agents and parasites. A talk about disease of albatross on Amsterdam Island by Amandine Gamble, and she will be today later on talking about this topic. And Jerome Faure gave a broad overview of ecotoxicology issues in Arctic seabirds, with showing us the importance of considering a hierarchy of scale, and notably quite broad scales for sampling and the importance of coordinating sampling at large scale. We had also a talk by Ray Grimaldi on human as vector of infectious agent in Antarctica, notably in relation to waste by humans, so very much related to the previous talk by Michelle Power, with issues of pollution due to bacteria that are brought to these remote places, as well as the wastewater management in polar area topic, which was addressed by Gwen Fenton. So I'll just give you a brief illustration of some kind of topics that are quite important with vertebrates in polar areas, notably South Polar areas. And this is related to work I have been involved in, but I guess this will give you a kind of a type of example of the kind of research and questions that arise. So vertebrate in polar areas, marine vertebrates, terrestrial marine that are on land, are exposed to pathogens and parasites, notably when they breed on colonies. Many of the species are colonial breeding species, like penguins here, or albatross below, or puffin kittiwakes in the Arctic. This is penguins and albatrosses are in the south. They can be exposed on these places because on these places where they aggregate in high density, they can transmit the infectious agents. They can also be exposed to vectors, such as ticks, that can wait for these birds to come back year after year and raise their chicks in this environment. So we have this series of species that are breeding very high density, coming back to the same areas where they can be exposed to the parasites and pathogens. This also goes for elephant seals or fur seals, which also aggregates. And we have also another group of species that can be especially interesting to consider in terms of the ecology of these host parasite interaction, which are scavengers and predators, such as this subantarctic skua on the top here, and below these giant petrels. They can be exposed to many infectious agents, notably when the birds they are eating have actually died of infectious disease, such as here an albatross that died of avian cholera on Amsterdam Island on the top. And they can potentially be dispersers of the infectious agent, but they could also be sentinels for this kind of environment because of their feeding habits, and could be used to track what kind of infectious agents are circulating. So this is just an illustration again of the fact that in this polar environment, a lot of the biodiversity, at least for vertebrates, is aggregated on these colonies of vertebrates, where the birds or the marine mammals can be exposed to ticks, directly transmitted agent, and this in relation to the high density of the colonies. Some issues that are maybe less known is the fact that a lot of these species are very long-lived, very site faithful, so they come back year after year to the same site, and this makes the interaction with the local population of parasites and pathogen perhaps quite strong and interesting to study. Notably, the immunity, well, the individuals are re-exposed to the same parasites at the same site year after year, and this can be quite useful even for studying some of these long-term interactions in the field. And of course, foraging ecology issues are also important because they expose a lot of the species to some pathogen and parasites. And for now, I've mostly been talking about what the animals are exposed to in the colonies. But of course, when they feed on fish or other animals, they can also acquire endoparasites that can also by themselves be very important to consider. And so these vertebrate population in polar areas constitute interesting study system in a context of global change, notably with a strong effect of climate change in these areas can have cascading effect on host parasite dynamic. I mentioned earlier introduced species, which can in these remote areas and relatively isolated areas can have very strong effects and can be very interesting and important to study. And in relation to that, the risk of emergence of disease can be tracked and can be actually occurring, notably in relation to human activities, as we have seen in the previous talk. So the type of questions that people are asking is the main one. First description, what agent, what parasites circulate in these areas, to which parasites individuals are exposed to, at what scales, only locally, or is it the same at a broad scale? We have seen that the seabird tick, Cetaceans, is really distributed at a really broad scale. In relation to that, what host movement may matter for the spread and the dispersal of some infectious agent. And on this, I would like to stress some of the work we did with seabirds, where we stressed that not only migration may be important, and actually maybe migration is really not so important in many of the seabird species, but maybe what is most important is what's going on the colonies in terms of foraging, prospecting visit by individuals between colonies, and dispersal of individuals, which can lead also to the dispersal of pathogen and parasites. What effects have these agents? This is not easy questions to answer. What drivers of these effects and what implication for management? We will see in some of the talks that this can be important issues to consider. And so the approaches are, some of them are relatively classical: field sampling, molecular serology analysis on the samples, with new tools getting available. But in parallel to that, when we consider ecological issues in the host parasite interactions, there are other approaches that are really useful to consider, such as ways to track movement of hosts, the genetic of vectors, the potential usefulness of carrying experiment in the field, and modeling of the dynamic of these systems. Just one mention about experiment, for instance, using in some very particular case when disease can really affect a host population that's threatened for different reasons. We discussed the use of vaccination as a tool for conservation in this kind of system. So among the many different things that were discussed, we realized that there is a strong need for more coordination among groups of sharing experience, integrating approaches, and training. Just illustrating here what we have set up in the French subantarctic islands, trying to train people in the different field days to be able to collect samples in case of a massive die-off, and training people in field microbiology, and of course conducting field work to gather key samples, which are key aspects of the work related to the ecology of host-parasite interactions. And one point, of course, that was discussed and raised by Andres Barbos, that was the importance of coordinating sampling protocols, which had been started earlier, but this is an issue on which we will carry on working as a group. And so this was a brief overview of that meeting. I think on top of stressing the importance of coordinating the type of sampling and coordinating the data that's gathered at the international scale, it was the occasion to stress the importance of integrated approaches, not only focusing on disease veterinary approaches, but integrating other aspects from population ecology to social sciences also, especially in the Arctic when there was issues about zoonosis, and the importance of long-term monitoring, as well as the importance of being able to ask original questions on this kind of system. So important questions about the effect of pathogen, but also indirect approach to issues such as shown in the talk before, the impact of humans in the remote places where they can bring things and they can, when we can track the impact of humans by tracking the infectious agent that they are bringing in this kind of environment. So I thank you for your attention and be happy to answer questions later on.
Perfect, thank you very much. So that's sort of the end of our first session. So we did have a couple of questions. So Rupert was looking at basically our main interest in looking at SARS-CoV-2 in Antarctica and sort of what we were sort of focusing on with that. So as Andreas has said, we were interested in looking at how we can avoid ways to avoid the virus getting down there and introduced, but then also trying to understand sort of what impact it might have on wildlife, and also whether there could be sort of that reverse transmission back to humans as well. And Michelle, we have a question for you, which is many national Antarctic programs are currently modernising their facilities, or at least planning to. How is this work that you do communicated to environmental managers and Antarctic treaties so they can make informed decisions about waste treatment?
Thanks, Natasha, for your question, and thanks, Megan, for reading that out to me. So obviously expert groups such as the one here with our panel members communicate back through those pathways. Obviously, scientific publications and hopefully the scientific evidence out there is being integrated in literature reviews and informing decisions. And also down to researchers to communicating the impacts and making sure it gets through to those channels. I don't know if Andre, who has a little bit more involvement with SCAR, might want to comment on that.
Yes. In fact, anything that can be discussed in the Antarctic community is informed by different ways. One way is the information through the national Antarctic programs to the Antarctic Treaty, and one of the other ways is through the national representatives in the different organizations. For instance, COMNAP. So there is a representative of all the countries that is involved in COMNAP, and there is, as far as I know, there is also discussion of everything that has to be with logistics, but also with the operation of the stations and the ships. And within the Antarctic Treaty, the organisms involved in preservation and environmental conservation is the Committee of Environmental Protection, and it's also the way that the national Antarctic programs communicate the information of what's important in the issues related with the conservation. And concerning the SCAR, there is also different ways that the researchers can communicate with SCAR. This is more open. There is also national representatives, but there is many different expert groups, working groups like our group, action groups that can make advice and communicate the results of the research to this organization. And the SCAR has a special link with the Antarctic Treaty because he's the scientific advisor of the Antarctic Treaty. So anything, any scientific results or scientific important thing that is communicated by the community, that the research community, will be communicated from SCAR to the Antarctic Treaty.
Fantastic. Thank you very much, Andres. So I'm going to hand over to Tom Hart now, who's going to chair our next session.
Hello, everyone. So yes, I think we're slightly over, so I'll start by introducing Michelle Villi.
Thanks, Tom. So I'm hoping everyone can see the screen. I'm just going to briefly talk to you about some of the knowledge gaps that we've got out there. Sort of full disclosure, I'm a virologist, and so the bulk of this talk is going to be about viruses. I've got 2 slides towards the end about bacteria, and there are a number of experts on bacteria here that can probably answer those questions better than I can. I'm not going to talk about parasites because there is a talk about parasites a little bit later on. So in general, we still have a lot to learn about viruses in birds, not just Antarctic birds, but in birds in general. The first viruses were described in birds in the 1920s. So we've been essentially working on viruses in birds for 100 years now. But the vast majority of viruses we know anything about are those that cause disease and those that are associated with poultry. It's really only in the last 10 years with novel methods that we've been able to better describe the viruses that are found in wild birds. And the majority of those don't actually cause any observable disease. So just quickly, you're looking at a plot here of sort of the number of virus species that have been described over time. And as you can see, a lot of effort's been done in birds. Prior to 1995, we already had over 125 viral species described. But as I said here, really only towards the end are we describing virus species in wild birds. In Antarctic birds, there's also been a really long history of interest in viruses. And so what you're looking at here is a figure from a review we did on avian influenza viruses in seabirds. And people started collecting samples to test for avian influenza viruses in the 1970s, the late 1970s already, and that has been ongoing. So this figure here sort of lists 3 different sample types that have been collected, and the colors refer to the number of samples that have been collected over sort of that 5-year interval. And so, for example, you can see in the 1970s, thousands of serum samples were collected from penguins. But sort of due to methodological constraints, it's really only in the last few years that we've been able to describe these viruses. And so our understanding of viruses in Antarctic birds has really been sort of colored by methodological constraints and how we look for viruses. So for the most part, if you review the literature, you'll find that the vast majority of studies are trying to investigate the presence of the viruses that are found on this sort of screen here. These viruses are important for poultry, okay? They cause disease, they cause us lots of money. But I wonder whether it is reasonable for us to continue looking for important poultry viruses on a continent where there is no poultry. And sort of when I talk about viruses in the freezer in the next slide, I'm really talking about these kinds of viruses. These are things that we know about, we've been studying for many, many years, and they're a reference to which we compare everything. So if we look at the history of sampling in Antarctica, sort of since the 70s and ongoing is largely the use of serology. Now, as Thierry mentioned before, serology is an incredibly powerful tool and very, very useful. And so essentially what you do is you go and you catch your species of interest, you take a blood sample, you spin that down, you get serum, and then that gives you antibodies. So you have birds that have antibodies, and to know what they have antibodies against, you compare that to viruses you've got in the freezer. So especially in the early years when we were doing serology studies, we would take blood samples and do antibody tests against things like influenza viruses, against things like paramyxoviruses. And sort of as a result, there are known viruses in birds through serological screens, but there are very few and sort of far between. So the first descriptions of things like avian virulence 1, this used to be called avian paramyxovirus 1, or Newcastle disease, a really important avian virus, and influenza virus were already described through serology in the 1980s. In the 1990s came infectious bursal disease. But other than that, there hasn't been a whole lot found. The other way that we have been trying to look at viruses in birds is sort of through virus isolation and later through molecular tools. So virus isolation is an extremely old technique. Essentially, you go down and you catch your birds, you take a sample, which may be something like a cloacal sample, and you inject that into chicken eggs, and you wait a few days, and then you harvest the fluid out of chicken eggs. And then you mix that with blood to see if there's something there. But of course, the limitation, especially sort of in the early years, pre-2010, is that what you found had to have characteristics of existing viruses so that you could place that into context of what you already knew. So again, useful, but has lots of limitations. Sort of in the mid-2000s came the advent of a tool called PCR. This is a very sensitive molecular diagnostic tool. This is what we're using to sort of identify COVID in people at the moment. But again, this is really limited to what we know, sort of viruses that we've known before, because it requires that we know the sequence so that we can sort of create and design these little sequence, these primers that match to sequences. So it doesn't allow you to identify anything novel. It only allows you to sort of describe what has been described before. And then in the last 5-6 years, we have sort of moved on to these new tools, which are called metagenomic and metatranscriptomic tools. And these tools allow us to stop comparing to what we've got in the freezer and start looking at what's new with fresh eyes. And so the result of that is we have this really strange history of virus description in Antarctica, wherein in the 1980s, viruses were isolated through this method, but they were quite different from everything that was in the freezer. And so we don't actually know what these things are, these viruses from the 1980s. I predict there are viruses called Evulovirus 17, 18, 19, which we actually formally described only 2 or 3 years ago. But those isolates have been lost. But here you can see a lot of effort and a lot of description occurring since 2005 with avian viruses. And if you look at 2020 alone, there's the description of sort of 17 or 18 novel virus species in Antarctic birds. And sort of with these metatranscriptomic tools, I predict that this number will continue to increase. Now, I would just like to sort of note that viruses do not necessarily cause disease. These are not necessarily pathogens. They're just viruses that are there, and it's important for us to sort of have this understanding of what is baseline, such that if we have the introduction of something that may be a pathogen, we know whether or not it's already in Antarctic birds. And so that's quite crucial. Now, sort of if we look at the viruses that we have described, which is not that many in the last few years, there are a number of important biases in the data. The first is that almost all virus description has occurred on the Antarctic Peninsula. Okay, and so that's mostly because the paper that has described the largest number of viruses was a study undertaken on the Antarctic Peninsula. There has been effort on other parts of the continent, but the most recent efforts have occurred on the peninsula, and whether sort of King George Island and the northern parts of the Antarctic Peninsula is reflective of the rest of Antarctica is really, really unknown. The second really big bias here is that almost all viruses that have been described in Antarctic birds are viruses from these 3 species of penguins. There has not been large-scale effort in things like skuas and things like giant petrels, which, as Thierry pointed out, could be extremely interesting because they're predators and scavengers, and they can move large distances. Furthermore, whether these penguins are reflective of all the penguin diversity, so the things like emperor penguins that are out there as well, is really unknown. I'd say the most important sort of shortfall and bias that is in the data is that what we've done so far are snapshot studies. So we go in, we collect samples, we describe what we have, and then we leave, and there's no follow through. There's no resampling. Samples are collected largely opportunistically, so the metadata might not be very good. We don't know if we've sampled adults or juveniles, males or females. We don't know whether we're sampling residents or non-residents. We may just sample one location, so we don't know whether what we found is reflective of other locations nearby, and what's happening over a period of time. And so that's a really big downfall. I say there is only one exception, and those are studies of this new virus, or 3 new virus species called avian Evulovirus 17, avian Evulovirus 18, and avian Evulovirus 19. And I think the fact that there's been a few papers on this is partially by chance, but partially because these viruses have been shown to be quite common. And so on the top right there, sort of data comes from these 3 publications. I've added that top publication up there as well because I predict some of those early viruses that were described, PV7, PV8, and 17, 18, I think it is, are these avian Evuloviruses, but I can never confirm that. I just suspect that. But essentially, through both virology and serology, these viruses have been found in Adelie penguins, gentoo penguins, chinstrap penguins. They've been found across different years. So the first virology study came from 2013. A big serology study was done in 2017. And another virology study came out with samples from intermediate years. So we know they're found in all these penguin species and they vary a little bit. We also know these viruses vary in prevalence by host age, and such that we found these viruses to be more common in chicks than in adults. This was statistically significant in one of the studies. And one of the other studies looking at serology also found differences across locations. And so this is sort of the thing that we need to start moving towards rather than just snapshot studies. I think those snapshot studies are really useful in helping us describe the diversity that's out there. But just because we found a virus in Antarctic penguins doesn't mean we know that it should be there, or how common it's there, or whether those penguins are reservoirs. So that's why we need these virus ecology studies as well. Just to briefly talk about bacteria, as I said, I'm a virologist, not a microbiologist, so there are people on the panel that are certainly more expert than I am in this area. But what I've done is sort of divided bacteria into 3 categories. And the first one is bacteria that are of concern to human health. So Michelle Power really started us off this morning on a talk about antimicrobial resistance and in E. coli, but antimicrobial resistance is important for people. It's important for us such that when we get sick, antimicrobials may not work, but does this have an effect on Antarctic birds at all? Just because it's there, is it doing anything? And the same can go for things like Campylobacter and Salmonella, okay? So early research into bacteria really started on these things because these are bacteria we knew something about and they were important to us. In terms of Campylobacter, the one that is commonly found in chickens and agricultural animals that cause food poisoning and more severe disease in people is called Campylobacter jejuni. It hasn't been found in Antarctica yet, but it has been found in the subantarctic, which is perhaps less surprising given the introduction of things like chickens to some of the subantarctic islands. But rather we see other species of Campylobacter in Antarctic birds, such as Campylobacter lari and subantarcticus. And there's a little bit of data sort of based on prevalence in different species and different locations, but studies are very haphazard and definitely snapshot and span a really large period of time, so difficult to compare. Salmonella was one of the first bacterial species that was worked on, again, because this is very important to human health. The way we've thought about bacteria has changed sort of from serovirus to species to sort of other things, so it's a bit difficult to understand how many species of Salmonella have been described as compared to serovirus, but there are a few important ones that I sort of put up here. They've largely been found in scavenging species, albatross and penguins across the Antarctic Peninsula. Many old studies, but few new studies. Antimicrobial resistance has already been covered well. I just thought to mention that we have found at least 4 classes of resistance in Antarctica. So genes against tetracyclines, beta-lactams, MLS, and phosphomycins. And in one study, they showed more AMR genes in penguins sampled close to stations that have sort of more action. There are more people there as opposed to more remote stations. The second category is bacteria of concern to avian health. And so Amandine is going to talk about avian cholera later, so I'm not really going to talk about it, only to mention that there is a history of avian cholera in both Antarctica and the subantarctic islands. They may be sort of linked through species like giant petrels. So early work by Leotta showed sort of an outbreak in penguins. They've also found it in, I believe it was a giant petrel, and it is known in the subantarctic islands, so whether those things are connected or not, Amandine will probably be able to explain better than I can. There is evidence of some other bacteria that may be of concern to avian health, but not well surveyed, not well recorded, very difficult to understand whether there is a direct connection or just sort of detection. And so there is a lot of opportunity for work there. The last category is sort of moving towards microbiome studies. So rather than looking for single bacteria, we can use technology to go in and describe the community of bacteria that are there, and then measure sort of how these communities change across sort of different treatments. And by treatments, I mean things like molting birds versus feeding birds, or adult birds versus chick birds, how these things change. Despite the importance and the sheer number of studies on microbiomes worldwide, I've only found 3 papers in the literature looking at microbiomes of Antarctic birds. This was far less than I expected, and sort of analysis depth of these studies is a bit superficial. The granularity of how they classify the bacteria is very high, so we're looking at very high orders and things like that, and so it's very hard for us to understand what exactly has been described in these birds. And I think one of the biggest things that I missed is that these microbiome studies don't link back at all to some of the previous studies looking at bacteria. So for example, we know that Campylobacter, through studies on Campylobacter are found in Antarctic birds, and how does that compare to sort of these microbiome studies? Is it something that's common or rare as compared to the rest of the community? So parasites, as I said, I'm not going to talk about. There's another parasite talk coming up. And so just to wrap up some of the thoughts that I have on some of the big knowledge gaps. So for viruses, what is the diversity of viruses in Antarctic birds? Which Antarctic birds are reservoirs for which viruses? How are viruses shared between places like the Antarctic Peninsula and perhaps South America, where we know there is sort of movement of birds? Do the viruses that we detect cause disease? How are viruses shared between different bird species? And sort of other ecological questions like age effects, location effects, seasonality. For bacteria, what constitutes the normal microbiome of Antarctic birds? Which bacteria are a disease risk? And this really would require going to species or even serovar level to identify those sort of disease-causing strains versus other strains, and what modulates this. What does the detection of bacteria of human importance actually mean? So detection of Salmonella, Campylobacter, E. coli, Klebsiella, and AMR, what does it mean? Is it important? Should we be concerned about things like that? And with that, I will take questions. Actually, questions later, sorry.
Thank you very much, Michelle. Yeah, if we can have questions at the session at the end, but please save your questions. So next up, we have Maria Leonardi to talk about a similar review, so the gaps in mammals.
Hi, are you listening to me? I have some problem with my internet connection, so maybe I turn off my camera during this presentation. Well, thanks of all. I really want to thanks to Andre for the invitation and to all of you to join us. I'm Soledad Leonardi. I'm working on the Institute of Biology of Marine Organisms, depending of the National Research Council from Argentina. And in this presentation, I would like to present a brief summary of what we know about sea parasites and what we are working on. Well, so we know global warming and its consequences constitute one of the many stressors for organisms around the world, affecting different factors such as the geographical distribution, the abundance, virulence of parasites and diseases, which in turn can affect the immune system of the host. In Antarctica, we have also considered the effect and impact of the overfishing and the raising of the human presence. All of this make evident the need to take some urgent measures as the creation of marine protected areas, and the need to have baseline information on immune parameters of organisms and the dynamic of host-parasite interactions in order to make future comparisons and to measure the impact of these stressors in the changing ecological context. In the sense, the possible effects of these changes include changes in food webs, changes in the habitat, the apparition of new diseases or zoonotic diseases, and all these changes in trophic chains and the reduction of sea ice could alter the current associations with parasites with an unknown impact on the health of the animals. In this sense, we expect a great impact of these changes on seals because the condition of top predators and CI-dependent species. However, we can assume that this impact will be different in each species in function of their habitat and CI requirements of each one. In Antarctica, there are 4 pinnipeds that are directly associated with the CIs, known as pack seals: the crab-eater seal, the leopard seal, the water seal, and the rose seal. I particularly not include information about the rose seal because we have not information about this species, but instead we'll mention something that we are working with elephant seals. Well, as I mentioned before, the magnitude of the impact upon each of the pika seal species is likely to be different, and in this sense, the species that depend on CIs for most or at least critical periods of their life story, or those that are kill specialists, should be more affected than species with fewer habitat requirements or with a broad spectrum. In this, considering this, we expect that the crab-eater seal, for example, will be the species most impacted by climate change-related alterations. Crab-eater seals are a highly ice-dependent species, and the diet is almost exclusively composed by krill. Something similar occurs with the whale seal, that is an ice-obligated species, but with a wider diet that includes fishes and squids. In the case of the leopard seal, we consider that it's a tolerant species that have the ability to adapt to use different ice flow types and explore a wide range of prey as fishes, seabirds, and other seals. And the same is expected to the elephant seal, that is a tolerant species that also reproduces on land. Okay. The Western Antarctic Peninsula pack seals have the highest density population within the circumpolar Antarctic sea ice, an area that also supports an important fishery pressure and receives the greatest number of tourists per year. In the sense, the need to monitor the health of the Western Antarctic Peninsula seal population is fundamental. But, however, there are little information available about parasites and diseases, and the information available is scarce and fragmented. We have little information on prevalence and health significances. Just for mention some of the most recent works, we have a work from 2014 from Regine Ferreira and collaborators, where analyzed the presence of some mean parasites in feces of seal from different locations along the west coast of the Antarctic Peninsula. And well, I suppose that all of you know the book of Kerry and Rydell, which includes different reviews of the health status of this species. And since 2014, in collaboration with the Argentinian Antarctic Institute, we start to work with some parasites. We start working with lice, and then we develop other lines that include other aspects of seal health that I will mention later in the presentation. And I want to present a brief summary of what we know about the species of ecto and endoparasite that infects seals. Let's see that for the 4 species, we have a description from some species of lice. Lice are specific, so we have 1 species of lice to 1 species of seal. And we have some species of Pacatocephala, cestodes, nematodes from the crab-eater seal, the fishes described from the water seal, and the leopard seal is just a quick review of this information. But this information allows us to identify some research gaps that include the need to increase sampling effort in some areas, to develop epidemiological studies along the time, to analyze the frequency, the prevalence, and some possible variations on the host parasite associations in this changing environment, the impact of these diseases and parasites on host health, and the need to continue with some taxonomy revisions because we continue founding new species. When we look for some old descriptions, we found that for the current standards, the original descriptions are incomplete or based in few individuals. And just to finish, I want to briefly tell what we are doing in Antarctica. We are analyzing some fecal samples, and we found some new species of nematodes. We're working in the redescription of some species of Corinosoma from elephant seals. We are conducting the monitoring of the health status of the seals, analyzing leukocyte counts and the presence of immunoglobulin G against canine distemper virus. In the sense, we found positive serology from all of the leopard seals analyzed, and the 90% of weather seals, but no one of the crab-letter seals presents positive serology for distemper virus. We also continue working with lice in some ecological aspects, but also we are analyzing the effect, the possible role of lice as vectors of some viruses or bacteria. And in this sense, and as was mentioned by Andres, little is known about viruses and other pathogens from Antarctic marine mammals. And we think that on the current context of global warming and the COVID-19 pandemic, the study of viral infection and associated biosecurity measures become fundamental. So that's all. Thanks for your attention.
Great, thank you very much to both speakers, actually. Don't forget there are questions and answers ongoing in the Q&A tab down below. So there was one question from Gary, possibly Gary Miller, about not forgetting things like sheathbills and kelp gulls migrating between South America and the peninsula as potential vectors. But thank you very much to both speakers. I think I will hand over to Rachel.
Thanks very much, Tom. So our next speaker is Ralph Venstrals, and he's going to be talking about parasites of polar wildlife. Over to you, Ralph. Ralph, it's your time to speak if you're able to. Oh, Ralph has been lost into the internet.
Yep, I've just got to fix him back up. There we are.
Apologies for the delay, everyone.
All right, you should be good to go, Ralph. You should be back in. Hopefully.
Hello, everyone. Can you hear me?
Yes, we can hear you.
Okay. Sorry about that. My laptop just shut off and I had to run for my wife's laptop. Let's try to share the screen. All right. Can you see my screen?
Yes, we can. Thanks, Ralph.
Okay. Well, hello, everyone. My name is Ralph, I'll be briefly talking to you about the parasites. Although I'm the one presenting, this presentation was actually prepared by 3 of us. So Daniel Gonzalez Acuña from Chile and Julia Diaz from Argentina are also responsible for this, this talk, and they'll be available to answer the questions as well. So the idea is to give you a brief overview of what we know about parasites so far. Oh, I can see the changing laptops ruined a little bit my formatting. Please bear with me. So why should we study parasites? Well, parasites are estimated to be about half of all the species on Earth, right? So that alone should convince you they're a massive part of biodiversity. When you look at the Antarctic land outside of the water, it's a desert really. There's not much biomass being produced. So if you want to make a living as an invertebrate, basically one of your only options is to go and be a parasite of someone else who's feeding at sea, because at sea there's plenty of resources. So especially in Antarctica, parasites are a large component of the life. Also, because they have such a complex network of hosts and the way they're transmitted through the diet or directly if they're external parasites or through vectors, they're very well connected with the web of hosts in the Antarctic. And this means that if you have changes in the environment, these connections will propagate, their effects will propagate throughout the trophic web, and this will change parasite dynamics. So if you want to understand a little bit about the host, you can learn quite a bit by looking at what's happening on their parasites. The population dynamics of the parasites is deeply connected to that of their hosts and vice versa. And you have also the environment is a large component that modulates this interaction. So in a healthy environment, these interactions are in a balance, in an equilibrium state. But if you get a drastic environmental change, say because of climate change or pollution or human presence, this whole system will shift and it might lose its balance, right? And in some situations, it might be quite detrimental to the hosts. And as well, if we look at this recent paper by a large group of researchers from SCA, they identified 6 priorities for Antarctic science, and 2 of them do connect to some level to the parasites. So learning about how Antarctic life evolved and survived. As I mentioned, parasites are a big part of life in Antarctica. So if you want to understand it, just in general, the species, but also what's happening with the hosts, it's very important to look at the parasites and to recognize and mitigate human influences. And as we'll talk, parasites can be affected quite a bit by what humans are doing in Antarctica. If we go a little bit back to the past, there were several stages in research of parasites. The first stage would have been the heroic age, when you had these expeditions in the 1800s and before the First World War. You would have these discovery expeditions, and you would usually have a naturalist on board. And this naturalist would be responsible for basically going out and hunting animals and collecting and bringing them back to the museums. Then from examining these museum specimens, we would get the first descriptions of the parasites. Because of the way the carcasses were preserved, there was obviously a bias in which kind of parasite get described there. So especially external parasites are really described during this era. And then we have a following era from the 1940s to the 1960s, which we could call a golden age of parasitology in Antarctica. This is a period where you have a number of university professors and also wealthy people who just take parasitology as a hobby, and they dedicate their lives to examining museum specimens. They have their private collections. There's more availability of expeditions to Antarctica. And so there's a boom in number of species described, especially for external parasites, but also for internal parasites. They also go through the effort of reclassifying and correcting the records that were made early. And they developed the first identification keys that, to be honest, we still use to this day. They might have been adjusted a little bit, but they did the bulk of the work of what we know about Antarctic parasites during these decades. Then in the information age, so before the 2000s, for helminths, there was a continuous growth. So the studies continued describing several new species. But for arthropods, with a few exceptions, we see a lot of taxonomic impediment, which means there was a lack of researchers. As the old researchers retired, There wasn't a good recruitment of new researchers. Entomology, systematics was not seen as a valuable field. It was not a very good career. And so it dropped off and we didn't get that many descriptions. Of course, we have some good exceptions. So for example, lice, there was lots of studies, feather mites as well. But most of the research during this period was about cataloging host parasite associations. So it was about expanding the geographic and this host species coverage and just re-recording the same parasites, but in different hosts or different locations. This was also the time when we had the first life cycle studies. So not just recording the species, but try to identify who does it infect, which fish does transmit it, and how do the hosts get it, and that kind of thing. And then we move to the present, which would be the 21st century. And we have this massive expansion in the quality of the logistics of working in Antarctic and the subantarctic. We're still struggling, quite frankly, with taxonomic impediments. So being systematist or parasitologist is not necessarily an easy career. There's a steep learning curve. There's not necessarily lots of jobs in this field. There's not necessarily lots of funding for this, but there are still some brave ones. And well, the first step that we've been trying to do is first to redescribe the parasites that we're seeing in the 40s, 50s, 60s, but this time redescribing them with genetics and with electron microscopy. So we can refine what's done and we can create a catalog of genetic information. So we know what, say, Tetrabotrys looks like genetically, and then we can use this tool to study the parasites more. There's also been a large number of single host studies or single parasite studies, where you choose, say, one species of host, and then you try to really dissect all the parasites that they have in one population or several small populations. This is the first attempts of establishing the epidemiology and the ecology of these parasites. And there's first attempts, we're just starting now, I'd say, to do multi-site collaborative studies, right? So it's not just one researcher in one site, but actually a network of researchers collaborating. We've seen as well the first studies on the effects of the parasites have on their hosts in terms of the disease that they can cause or not, the decrease in fitness, things like that, and how do the parasite infestation correlates to environmental and behavioral characteristics of the host. And as well, we've seen the detection of vector-borne parasites in the last 2 decades, right? So because now we have molecular tools which are far more powerful in that sense, we've been starting to find protozoan and bacteria and viruses that are transmitted by parasites. And then we can study the second layer of this interaction. Things become increasingly complex the more we look. And there's been also lots of reevaluation and consolidation of past knowledge. So because we had that boom in the 60s, and then we had a bit of a gap depending on the parasite group. In these few decades now, what we're trying to do as well with the help of the internet, which allows us to do better literature searches and easier, at least, if not better. We can really recompile all the information that was done so far and then critically look at that and try to find which are the gaps, just like Michelle and Soledad did just about now, and reassess where we can go with our new tools and with our new insight. The future of Antarctica, we don't really know what it looks like. I doubt it will look like this picture, but we can think of some of the gaps that we have in terms of knowledge. So there's definitely a bias that we have in terms of the hosts that we have to research. We haven't studied all the hosts equally. So there's been a lot of study on the fishes, penguins, on the large sea birds, such as penguins and albatrosses, and on the true seals of Antarctica. Maybe because they're easier to catch, maybe because they're visible, maybe because they form large colonies that are more readily accessible. Whereas other hosts, such as skuas, gulls, terns, cormorants, petrels, all the small ones, the ones that make nests in burrows or in really inaccessible cliffs or things like that, those ones have been understudied over time, and fur seals as well have not been thoroughly studied. So maybe in the future, something we can focus on is trying to get a sense of these new hosts that we know so little about. We also have major gaps in terms of the parasite groups that are studied. So for logistical and practical reasons, it's easier to study large gastrointestinal helminths or lice, feather mites, ticks. So we really know a lot about these parasites. And comparatively, we know very little about smaller helminths or helminths that don't parasitize the gastrointestinal tract, but maybe they're in the respiratory tract or in the kidneys or in the nasal passages. Nasal mites, nest mites, tongue worms, which are very primitive kind of parasite that infects the respiratory tract of some birds, and the vector-borne parasites, because we had a limitation in technology that we now can finally address. So as well, maybe we can, if we focus more effort towards these parasite groups, I think there's still a lot to discover. And there's definitely, we don't know much in terms of epidemiology and distribution of these parasites. And we also have major geographic gaps, right? So a lot of what we know comes from very few places that for logistical reasons had better set up Antarctic bases or logistically were easier to go to. So South Georgia, the Antarctic Peninsula, the Ross Sea, the Kerguelen Islands, Macquarie Island, those locations were very thoroughly studied, whereas we have some areas that were studied to some extent, but we still don't know a lot for some of the other parasite groups or for some of the hosts. And then you have these many places that are complete dark spots. So Bouvet Island, the South Sandwiches, Scott and Balleny Islands, Peter First Island, Diego Ramirez Island, and large extents of the Antarctic mainland that there's been virtually no studies on the parasites. I would suspect we will find the same species, but because the communities of hosts are so different, the epidemiology, I would expect, would be quite interesting. And now that we are slowly building a library of genetic sequences for these parasites, we can start using them to study non-invasive samples. So for example, we can go to a colony and collect feces, and then we can look for DNA, and we know what DNA of Contrasecum look like, what DNA of Cosmocephalus look like, Tetrabotrys looks like, and we can, with that previous information, we can decode the DNA that we're collecting in those samples. We also now with DNA, we can identify stages of the life cycle of the parasites that would not be identifiable before. So cysts and larvae that don't have enough characteristics to identify the species. With DNA now we can do it. So this opens quite a large field for us. And there's lots to do in terms of using genetics to study evolution and population dynamics of parasites. So understanding how the parasites are moving from island to island, which hosts are carrying in which direction, and well, just so many questions that are so interesting. The big gap of knowledge that we have very little knowledge about is, well, the diet will influence the parasite that a host has, because lots of the parasites are acquired through the diet that you eat. And also it will affect the immune status. So if you're well fed or if you have a specific content in your diet, you might have a better or worse immune system. And the immune system is directly fighting the parasites, right? So you have these three components that are interacting, and we don't quite understand how they interact. So we don't understand what does a food shortage do for which parasites, right? We don't understand what specific parasites do to the immune system, right? And which parts of the immune system are more important for different groups of parasites. So this is a huge field of research that we still have basically to start. Understanding how the parasites are interacting with each other, because they're all competing for the same host. And in some cases, they might find synergy. So if they figure out a way to suppress some parts of the immune system, then the other one can take benefit of that. But also they might be competing for resources in some cases. So there's lots of complicated ecological dynamics that we know from tropical and temperate environments that are probably also going on in the Antarctic and we know nothing about. And we have to study, we've been studying only at the population level, really, whereas we need to start understanding how it goes in the community level, so how different species within the Antarctic system are interacting, and then at the ecosystem level. So that would be understanding how a parasite from the penguin is transmitted or not to an albatross and to the fish, and then how all of this is connected to what's happening with the sea ice, right, with the tourist presence, with climate change and all of that. And then, of course, yes, the cherry on top of the cake would be to understand how climate change is affecting all this, because we know climate change will have drastic impacts in Antarctica, especially in West Antarctica and in the peninsula. And so want to understand how these networks of interactions in the Antarctic ecosystem will affect the hosts and the conservation of that environment at the end. Of course, we don't do this kind of thing alone. So we have lots of organizations that back us up and that make all of this possible. So of course, we would like to thank all of them. And that's it. Thank you.
Thanks very much for that great talk, Ralph. Megan, we are running a little bit early. Are you happy to continue with Amadine's talk, or do you want to?
Yeah, no, I'm happy to continue, and then we can. We've got 2 more talks, and then we can have a bit of a discussion.
Fantastic. So I'd like to introduce Amadine Gamble, who's going to be talking about avian cholera in subantarctic wildlife. Thanks, Amadine.
Hi. Let me know if you cannot see my presentation. Otherwise, I assume it's good. So yeah, thanks. Thanks for the introduction, and thanks for the opportunity to present our work. So yeah, I will be talking about avian cholera in subantarctic seabirds, and more specifically focusing on how combining epidemiology and ecology approaches can help us to identify potential mitigation strategies for diseases that are impacting polar wildlife. So before starting, I want to acknowledge that this is a team effort, and that's notably some work I've been conducting during my PhD with Thierry Boulinier, who presented earlier today. And we also have some other students and field assistants that have been really critical to the success of this research project. So avian cholera is not something that is super rare. It's not something that is typical to Antarctica. It happens a bit everywhere. It's quite famous for in poultry farms because it can lead to dramatic mortality that can obviously impact the functioning of the farms. It can also impact wild species, and there have been notably some outbreaks that have been relatively well studied for wild outbreaks in the Arctic on common eiders. And so you can see, for instance, on that picture, the kind of scene that we can see in the case of avian cholera outbreaks, where you will have thousands of birds that can die quite quickly. And also, so avian cholera is caused by a bacteria that is called Pasteura multocida, and this bacteria is not specific to birds. So I just wanted to highlight that it's a conservation issue for birds, but it can also be a conservation issue for mammals. And for instance, there has been a big Saiga antelope die-off that has been linked to Pasteura multocida, so the same bacteria that is causing avian cholera. My work was mostly focused on Amsterdam Island. So this island is located right in the middle of the Indian Ocean between Africa and Australia. And it's a really interesting system to study this disease because their outbreaks are recurrent, which is not something usual. Usually you will have one outbreak that can last for maybe a few years, but it usually fades out, and then the population seem healthy again, which is not the case on Amsterdam Island, where so doing seabird demography noticed quite sudden and massive mortality events affecting the nestlings of yellow-nosed albatrosses, as you can see on this picture, with like almost all the nestlings of the year dying within a few weeks. And this started in the mid-80s. And since the mid-80s, these events have been occurring every few years. And recently, they have been actually occurring every year. So it seems to affect only the nestling and not the adults. So the impact on the population might not be that critical at the beginning of the epidemic, but as it's been recurring for years now, there is actually a decline of the yellow-nosed albatross population of Amsterdam Island that has been recorded. So the bacteria Pastora multocida was first identified on this island, actually 15 years after the first outbreak. So in the 2000, just using some classical bacteriology tools. So it's quite tricky because you have to find a dead bird and to isolate the bacteria from it. And there is lots of competition with scavengers on the island. So it's not that easy to find dead birds, even if thousands of them are dying within a day. And so to give you an idea of what's happening within a season, you can see on the graphic on the left, at the top, you have the prevalence of the bacteria in fecal and cloacal samples collected from birds within a breeding season. So from hatching to fledging of the chicks, detected by PCR. And so you can see that you start every year with a relatively low prevalence that increase really quickly and reach up to 100% of the chicks. And this is like mirroring actually the nestling survival probability that you can see drops to null some of the years in parallel to this increase in the prevalence of the bacteria. So it's always difficult to be sure 100% that one infectious agent is causing a mortality event in a wild population, and even in domestic populations or humans. We are pretty confident in that case because we used a vaccine that was specifically targeting Pastora multocida, and that increased the survival of the nestling. So that's why you can see on these two plots. So on the left plot, you have the immune response following vaccination in vaccinated nestlings and non-vaccinated nestlings. And on the right plot, you have the corresponding survival probability of the nestling. And you can see that the vaccinated ones, half of them survived, while for the non-vaccinated ones, almost none survived. So this field experiments allowed us to demonstrate the causal link between Pastora multocida and nestling mortality, and also was opening avenues for potential mitigation strategies. But so it sounds like finding a vaccine can solve everything, especially in the time of SARS-CoV-2, for instance. But there are some logistical constraints, both in human populations and obviously even more in wild and remote populations. And notably in this case, we cannot imagine vaccinate all the chicks every year because there are thousands of chicks. The population is on the island is regrouping 23,000 of breeding pairs. So that's lots of nestling to vaccinate. And we would have to do it every year. So we are exploring other potential options. And I will present some of the ones we have identified. So why do we even want to think about potential mitigation strategies? Because that's not obvious. We could consider that this is a natural host-parasite interaction, and we shouldn't interfere in that. In that specific case, there is some concern because yellow-nosed albatrosses are a threatened population, and 2/3 of the world population are on Amsterdam Island. There is also a potential risk of spread to other species, and notably the endemic Amsterdam albatross, which has only a very few breeding pairs. So it can really easily disappear. And also, there is a bit of suspicion of human responsibility because we think that there might be, it's really hard to prove, but there might be an anthropogenic origin of the bacteria on the island. Notably, as in the past, as Michel mentioned it before, it was really frequent on subantarctic island to breed poultry and swine, notably, that can carry Pastora multocida. And they also introduced rodents on the island that can also carry the bacteria. So it seems like it might be a little bit of our responsibility to at least consider what is possible to do. And also from a really basic point of view, it's just really interesting to try to understand why Pastora multocida outbreaks are recurrent on this island, while they are not on other in other populations that have been subject to avian cholera outbreaks. So just to like frame the concept, like the context a little bit wider. So if you consider an epidemiological dynamic in an albatross population, you would have first an the emergence that can be caused by a spillover from another species or the introduction via contaminated material linked to like research activities or tourism. You can have mutation of the host or the parasite and so on. We can or not observe these events. And then you will have transmission from individuals to another. And this transmission can occur at different scales. So it can stay within a colony, or it can reach other colonies and potentially other islands. And as a pathogen will spread in the population, you can imagine two different scenarios, although it's hard to put a clear threshold between one another, but you can have the establishment of the infectious agent as endemic in the population, so it will stay in there and occur recurrently, or it can fade out. So that's a thing that we are kind of observing on Amsterdam Island. It seems that Pasteurella multocida is more behaving like an endemic infectious agent, while in other systems it has been more subject to phase out. And so understanding how the epidemiological network works is really critical to design appropriate and efficient mitigation measures. Because for instance, if you eradicate the pathogen from one colony, but there are actually recurrent reintroduction events from other colonies or via another species, then that would make your first eradication that was successful fail within a few years. So it's really important to identify the potential source of emergence of the infectious agents and the routes of transmissions. And as you can see in the example that I gave below, there are actually lots of these concepts that depends on elements that are external from the albatross population itself. So if you have spillover introduction, it means that you have another species that has brought the infectious agent in your system. Regarding transmission, you can have direct transmission, but you can also have environmental transmission, and as illustrated before, you can have vector-borne diseases, or you can have transmission of infectious agent via other species that are also infected, even if they are not your obvious subject of etude. So it's important to look beyond the obvious target host. So in our case, the first obvious target host is the yellow-nosed albatross, because it's the one that is obviously impacted by the disease, and it's also the one for which we have conservation concerns. So if we want to understand the intercolony transmission of infectious agents, and in that specific case, Pasteurella multocida, it can seem like, I don't know, almost easy, but it's actually not. Because seabird populations are really highly spatially structured, so they will form colonies, and there will be relatively few exchanges between the colonies. So it's not obvious that if you have an infectious agent in a colony, it will be able to spread to another colony. On the other side, we have extensive knowledge on movement of seabirds, and we know they can, for instance, travel for really long distance, and things like that can potentially impact the transmission of infectious agents at different spatial scales. So regarding seabirds in particular, the thing that makes it not obvious that movement can contribute to the large-scale spread of infectious agents is that most seabirds migrate and forage only at sea. So they spend really either not much time on land, and while actually they're on land, they are usually on their nest, and there can be very few interactions with conspecific or other species. They're basically just traveling back and forth between the sea and their nest. So this may limit the opportunities for transmission of infectious agent, because there will be not much contact between individuals. Like at sea, there is likely not much contact between individuals either. But so as Thierry mentioned it before, and some other people mentioned it too earlier, all seabirds are not behaving the same, and notably some seabirds also forage onshore. And that's notably the case of giant petrel skuas, sheathbill, and gulls. And they tend to be neglected, as it was pointed by Ralph and Michel. I guess it's because there are usually non-classified as threatened species. They are less emblematic, and also they can be less impacted by the infection. And actually, it's really hard to say because they are understudied. So even if something was impacting them, we might completely miss it. But because they spend more time on land, and because they forage on land, they might visit different seabird colonies and contribute to the spread of infectious agents. So we wanted to investigate that on Amsterdam Island regarding avian cholera. So we sampled some skuas, and we detected actually in almost all of the skuas with samples antibodies against the bacteria. So it means that almost all the individuals had been exposed to the bacteria at least once in the past. Some of these individuals were also actively shedding the DNA of the bacteria, so they were potentially infectious. So we have these birds that are potentially infectious and that are likely to visit other colonies. So to dig a little bit further, we put some GPS loggers on them, and with a strong expectation that, as you can see on the picture I put on this slide, that they will spend lots of time in albatross colonies because they actually forage on dead albatross. So here are the maps of habitat use of the breeding skuas on Amsterdam Island 2 consecutive years, and you can see that they spend lots of time on the southwest cliffs, which is where we have been monitoring the avian cholera outbreaks for the past 30 years, or avian cholera outbreaks have been recorded the past 30 years. But they also visit other colonies, so they can potentially move the bacteria from one colony to another. And it's even more impressive if you just look at the movement of some individual within one day. As you can see on this video, so it's probably really slow for you with the connection, but each color is an individual, and you can see that within a day, they visit several, like they stop at several colonies or several points where there are actually colonies of seabirds and marine mammals. And they also all have this stopover at the avian cholera hotspot. So there is this opportunity for transmission, but also super spreading, because these birds that are actually a small population on Amsterdam Island, it's only 60 pairs, so it's ridiculous compared to albatrosses. They can disproportionately contribute to the spread of avian cholera. And if we look, have a look at other species, so we also considered giant petrels and leather sheepbill that we sampled from other islands, and so notably Crozet, Kerguelen, and Marion Islands that are also in the southern Indian Ocean, and the Falkland Islands and in Antarctica. We got some serological data from these birds, and we detected actually antibodies in a high proportion of birds from all the islands of the southern Indian Ocean. So it means that potentially either there are other hotspots of avian cholera in the southern Indian Ocean, or these birds are directly or indirectly in contact with some birds from Amsterdam Island. For instance, if skuas migrate to the same spots in winter, or some skuas might actually migrate from the southern islands to Amsterdam Island. So it raises some questions whether to which extent the bacteria can spread to other islands. And investigations are ongoing, so for now we don't know really much more than that. So that was like considering another species with a like completely different behavior, but I also wanted to highlight that even within a species there can be some heterogeneity that can make some individuals more prone to spread infection than others. And Chiri mentioned this like earlier today, that depending on the life stage of an individual, they can exhibit different behaviors, and notably non-breeding individuals can prospect, so visit different colonies. It's thought to be behaviors that help them disperse. It has been overlooked in albatrosses because they are highly phylopetric species, so they do not disperse much so it was thought that they won't prospect. But we decided to investigate that anyway, and we actually realized that there is a non-negligible proportion of failed breeder and pre-breeders that do prospect. So what you can see on this map is breeding individuals that just, when they are inland, they are just in their colony where their nest is during the chick rearing period. While failed breeders and pre-breeders who don't have a nest, they tend to visit other colonies around, and it can actually be quite far. And yeah, they can like visit completely isolated, otherwise isolated colonies, and that also might represent an opportunity for pathogen transmission. And if you look into what one individual can do, you can also think that there might be some opportunities for super spreading, because here you can see all the red points correspond to stops that one individual, in that case pre-breeding individual, did during the tracking period. And so you can see that this one individual visited lots of other colonies. So identification, the identification of these individuals that can potentially contribute more to pathogen spread can help us to identify actually priority targets for mitigation measures. And you might have heard the same concept for SARS-CoV-2 with the chopping the tail strategy, consisting on identifying the activities that are linked to bigger clusters and just focusing on this one instead of trying to isolate everyone from one another. So for instance, we could imagine if we can identify a vaccine that is blocking transmission, we can imagine vaccinate skuas, and that could help reducing the spread of Pastora myrtocida on the island, or treat skuas with antibiotics or things like that. And that's a potential high benefit and low cost strategy, because you only consider a few individuals compared to the large population of Zebra species. And so there is still this mystery of why avian coral drugs are recurring on Amsterdam Island, while most of the seabirds migrate at sea during the winter. So there is no more host for the bacteria on the island during the winter. So there can be potentially, you can imagine that the bacteria is staying alive in the environment for a few months, which there have never been any detection of the bacteria in the environment, neither in this system or in other white systems. So it's not clear that it's actually something that could occur, but we haven't completely ruled it out. But there is also another species that is actually resident to the island that we haven't mentioned, actually two other species on Amsterdam Island, which has introduced rats and mice. And so they interact with seabirds, and they can transmit Pastora myrtocida from what is known in poultry farm. So here is an illustration of how they interact with seabirds. So they actually do not only scavenge, they can also interact with live chicks. So they could potentially contribute to the transmission of the infectious agent on the island. So we started to investigate that, and that's ongoing. So we sampled some rodents, we took some oral swabs, and we tested them by PCR. And you can see the red points are the PCR positive rats, and the yellow ones are the negative ones. And you can see there is this big cluster here where there are avian coral outbreaks in the birds. So that's probably rodents that are feeding on either dead or sick birds and get infected. But so if you want to have a closer look, that's what's happening precisely at this site. Where it becomes interesting is that if you look what happens in winter, so when there is no more seabirds at all in this area, because there is, all the seabirds that are breeding here are all migrating at the same time. The rats there are still some rats that are positive by PCR for Pastora myrtocida. So this highlights that potentially rats could contribute to the maintenance of the bacteria on the island. And so in that case, beyond the maintenance, they can also transmit the bacteria by biting the birds. And so that could be another priority target for mitigation strategies, and especially because there are lots of focus on the control of introduced species, and there are lots of islands that are planning to remove or have already removed introduced species from the island. So if rats are indeed involved in the maintenance and transmission of Pasteurella mucosida on Amsterdam Island. It means that removing the rats, in addition to the benefits that you can have from by removing competition and predation on the seabirds, you would also remove the potentially part of the infectious pressure that is exerted on the birds with Pasteurella mucosida. So overall, my big conclusion, I guess, are the same than everybody else before, is that we should not only focus on a few penguin species and a few albatross species, because even if that's your main interest, some other species can play a really important role in the processes that are impacting these species. We should also not neglect small population, because it's really not the size of the population, but the behavior of the species that can impact, or the species or the life stage or so on, that can impact their role as a spreader of infectious agents. And also that it's really insightful to coordinate together epidemiological and ecological studies to understand and potentially imagine mitigation strategies for infectious diseases in wildlife, in polar wildlife in particular. That's it for me. So yeah, thanks for your attention, and I take any questions.
Fantastic, thanks, Amadine. The next talk is by Megan Dewar. She's going to be talking about using genomics for disease surveillance. And if you have any questions just for this session, please put them in the Q&A and we will address them after Megan's talk. Thanks, Megan.
Perfect. Hopefully you can all see my screen. No, hear me? All good, perfect. So we've had quite a few talks today talking about our knowledge, or most importantly, I guess, our lack of knowledge when it comes to diseases in our polar wildlife. But also we've sort of touched on some of the limitations that we have had in the past in terms of the methodology that we've had to look at diseases in wildlife. So today I'm going to talk about some of the ways that we can use genomics to actually help us to look at diseases and so forth in wildlife. So as we know, around the world, diseases in wild animals are becoming increasingly recognised as a major problem for wildlife. And it is predicted to become a bigger problem as we get changes within our climate, but also increases in encroachment on wildlife from people. And the polar regions are not immune to this. So we are having increased human activity down in our polar region. So we have you know, people moving down there. So we have researchers, we have tourists coming in to support those people down there. We have a lot of infrastructure being developed. A lot of landscape changes are occurring, not only because of human activity, but also because of changes in the climate. Within our subantarctic islands, we are getting a lot of introduced pest species, but also there are potential risks of introduced species if we do get changes within our climate and so forth down south, especially around the peninsula area. We have increased human-wildlife interactions, and all of these are having an impact on wildlife health. And so it's not just in terms of introducing potential new pathogens to the region, but it's also potentially increasing their susceptibility to pathogens and other things that will affect their health. So increases in pollution and so forth, but also increases in stress and so forth, having people down there. And one of the big limitations we have is how do we monitor, how do we detect, and how do we investigate not only infectious diseases, but also native and non-native microbes and disease agents that are in the polar region. One of the big issues we have is Antarctica and even our subantarctic islands are quite remote. And so we don't have, you know, large laboratories and things nearby. So historically, in terms of disease investigations down in the Antarctic, a lot of things were done using traditional culture techniques, direct microscopy and cell culture. This was predominantly because they were the methods that were available at the time. A lot of these methods can be quite biased and limited. And the reason for that is, a large number of bacteria can't be cultured. It's not because you can't grow them, it's generally because we don't know what conditions they need to grow. But also, even your non-selective media is actually very selective, because there are some organisms that need really specific types of environments to grow. And so it can actually limit our ability to identify what might be causing a disease, but also just to do general surveillance and stuff of a wide variety. It can be time and labour intensive. Anyone who's worked in a microbiology lab knows that growing these organisms and then doing all of the biochemical tests and everything to identify your organism can take quite a long time. It can, of course, delay detection, and sometimes it can be difficult to identify the original source. As Michelle highlighted, a lot of stuff that has happened has been very sporadic, so we don't really have any ongoing surveillance programs, and most of the research has just been done sporadically by people who are interested in this field. And when it comes to pathogen work, a lot has been focused on detecting specific groups or specific pathogens. So generally a lot of work has been done on those human important pathogens that we believe might have been introduced by humans. And of course, some focus has been on things like avian cholera that we do know has had an effect. But there's still a lot of information that we don't know about. This is just an example of some of the things. So one of the big things we also don't know anything about is what naturally occurs within these animals, and therefore, how do we identify if we have inadvertently introduced a new microbe into these species, or what is pathogenic. And again, a lot of those historical studies used those traditional techniques, which doesn't really give us a very wide variety or ability to identify. So many of these studies pretty much identified that a lot of penguin species were colonized by only 1 or 2 microbes, and some of these studies identified that there are no anaerobic bacteria and so forth present. So as technology has advanced, our ability to do more advances as well. So as I said before, one of our biggest limiting factors for being able to diagnose diseases, but also understand what is happening, is not only our lack of information on etiology and the disease itself. So as Michelle pointed out, we have identified there are some human important pathogens in Antarctica and in Antarctic wildlife, but we still are yet to determine their impacts on the animals as well. But also, as I said before, understanding what constitutes the normal flora, what is naturally there. And unfortunately, we haven't really had a lot of these studies happening. And we also need to look at, where individuals could actually be exposed. What are the potential sources of infectious agents? So where could they be coming from? Could they be coming from migratory species? Could we be inadvertently bringing them in, not only ourselves, but on our clothing, our shoes, in sewage and so forth? So where could these diseases come from? And also whether or not these organisms can actually survive in Antarctica as well. And so one of the big things is, if we want to truly understand about wildlife health and their diseases, we also need to know what naturally occurs, not only in the animals, but also in those that they're interacting with, and what factors influence the presence. And so genetic sequencing is a method that gives us a few more opportunities to start to explore what is present in organisms. So it allows us to identify organisms either in pure culture or within mixed environmental samples based on their unique genetic sequence. So traditionally we had methods such as PCR, real-time PCR, reverse transcriptase PCR, which is really great if you know what you're looking for and you just want to do a presence-absence, or you maybe want to see if there's been a change in the abundance, or if there's a specific gene that you're interested in. So you might be interested in the expression of particular immune genes or virulence genes of a pathogen. But unfortunately, those traditional sequencing techniques don't enable us to detect differences in mixed samples. So, you know, you're very specific with these in terms of your primer that you have, and it will just say if it's there or if it's not there. It won't actually tell you, you know, what other organisms are there. And that's where a new generation of sequencing has come into play, probably over the last 5 to 10 years, but more recently for wildlife studies. And that's our genetic sequencing technologies. So these are very wonderful, powerful tools that we have. And these are basically quite large sequencing machines, but they basically are able to, depending on which method you use, you can actually start to look at the community that is there, not just one or two particular species. So it gives us the ability, we can monitor and detect and identify infectious agents. We can characterize the actual microbial community. So we can start to look at who is in there, who naturally lives there, and we can actually monitor changes of that composition and start to look at what's happening and whether things are changing. We can sequence the entire genome of pathogens, which is really important because that can give us the ability to identify important genes that they may have. We could maybe identify antimicrobial resistance. You can also start to look at where the infectious agent came from. You can start to look at the genetic similarity of strains in different places. We can identify the virulence and pathogenicity, and also look at the gene expression, so either of the host or of the pathogen. So these sequencing technologies have really revolutionized disease surveillance and also looking at our microbial world. And depending on what your question is, there are a range of different methods you can use. So you can be very specific and just look at a particular group of organisms. So you do your amplicon sequencing and just look at you know, the bacterial community, or you can expand that out to things like shotgun sequencing, metagenomics, where you can look at sequencing all the genes that are present within that mixed sample. So therefore you can actually identify bacteria, parasites, viral DNA, some host pathways, so genetic pathways and so forth. And then you can also do your RNA-seq and start looking at those gene expressions and your viruses. So benefits of these types of methods for the polar region, it starts to help us to build on our knowledge of what naturally occurs. So what is there? We can start to actually look at that whole community. The other benefit too is we can start to look at old samples as well, so historical samples, as long as they've been preserved correctly and the DNA or RNA is still viable within those samples. We can actually also start to trawl through freezers and start to get a historical perspective. And that's something that I'm currently working on with the Australian Antarctic Division. So we're actually raiding their freezers of samples over the past 20 years. And we're going to be using shotgun metagenomics to actually look at the microbial community and look at whether or not there are pathogens in those samples and how they have changed over time. We can use these techniques for ongoing surveillance and monitoring. So by regularly collecting samples and bringing them back to the lab and screening them, we can start to look at, have we introduced a new microbe or other microbes? We can do surveillance of migratory species. and starting to see if they're potential sources or vectors of introductions. We can also expand this out and start to look at other potential sources. So we can look at whether we ourselves are potential vectors, so really important for our biosecurity side of things. We can start testing sewage. So this is something that's happening in different places around the world for COVID. So sewage monitoring is actually being used for that, but we can also use it for understanding what we're potentially introducing into the environment. And as Michelle covered, with the antimicrobial resistance genes from the sewage testing that she did. We can also test our different biosecurity measures in terms of how we're cleaning our clothes and our shoes. So we can actually use genetic methods to see whether or not we are actually doing a good job and we are removing those pathogens, because a lot of previous studies were done using traditional culture techniques. So these are things that we can start to use not only to look at diseases in the birds themselves, but also to enhance our biosecurity and our monitoring and ensuring that we're not getting pathogens, or look at ways to reduce potential pathogens that we're introducing. We can also identify new diseases or pathogens that are showing up. And as we went through before, we can actually start to look at the pathogen genomes or even partial genomes and start to get some more knowledge on the characteristics of the pathogen. We can sometimes do tracing and looking at where the pathogen has come from, so looking at similarities in the genes and so forth that can help us identify what might be introducing those pathogens. But it also can help with monitoring outbreaks and pathogen spread as well. So, these genetic techniques, when we especially don't know what we're dealing with, can actually get us an ID a lot quicker than our traditional techniques that we use with culture. So some of the studies that have been done over the recent years, so there's been a few studies that have been coming out looking at profiling the microbial community, so trying to understand what is there, trying to understand what factors naturally influence that microbial community, so how does physiological processes such as fasting during molt influence that community, and also looking at whether or not pathogens are more present in those birds or not. Looking at different age classes, but also we've started to use these genetic techniques to give us more insight into our viral community. It's definitely something that is understudied in many wildlife species. We've been able to identify novel new viruses, as Michelle went through, and we've also started to identify some pathogens that might be present. And so this gives us an opportunity to sort of broaden our scope in a sense. So you can use these technologies to identify potential new pathogens, and then you can do a combined approach of genomics and traditional culture techniques to learn more about the different pathogens that could be present in these animals. So it actually broadens our understanding of what's there. This is what some of the data can look like when you get it out. So this is some work that we've been doing as part of our penguin microbes project. So this is using shotgun sequencing. And so you can actually start to see all the different microbial groups. So this is just down to genus level. And so we can start to see if there's species differences within species, or is there a core penguin microbiome or a core seal, or do they have differences? We can start to identify, those potential pathogens again. So as Michelle said, with Campylobacter, we can actually, look at, yes, we know it's a pathogen. Yes, we know we've detected it before, but in the scheme of things, how prevalent or abundant is it within the gut microbiome of those species? So we can actually start to get a lot more information. Like all methods, there are some downsides. It can be labour and time intensive. You need to be able to extract your DNA. You need to be able to preserve it appropriately. And then you need to prepare your library preps for sequencing. It often requires large specialised laboratory space. So these are some of the devices that are available these days. So this is this big baby here. This is a NovaSeq. So this is one of the very large systems that they have for really in-depth sequencing. That's probably about the size of a large commercial photocopy machine, or even the size of your washing machine. So it's a big machine. These other ones here are usually benchtop machines, but they still are quite large, and they often need sort of proper setups and specialized setups for them. It's often quite expensive to do as well if you're not doing it at a commercial scale. So the reagents and so forth can be quite expensive. So most people do get their sequencing done commercially. But also you then get a large amount of data. So some of these machines are pumping out, terabytes of data. But usually, from a project, you might get 5 to 10 gigs of data from your project. And it doesn't come out in a nice little package like this for you. It actually comes out as just, the genetic code, the genetic sequence, and you need to use specialized bioinformatics to analyze a lot of this data. Also, as you can imagine, you cannot do this in the field. So there is some time delay in terms of getting results back. So for just ongoing monitoring, surveillance, you just want to see changes in the community. If something's being introduced and so forth, then that's fantastic. These systems are very, very helpful for that. But in the fact of once you have a disease outbreak, that can be a little bit more difficult. So there will still be sort of that bit of a time delay in getting results. But the data that we do get from these systems are really, really good. And so because of that sort of limitation, because of these big machines and so forth, there are some new tools coming out that are providing us with more field-based remote opportunities for doing sequencing in the field. And so you have things like the bento box, the MinION, mini PCR. And so these systems are nice, they're small, they're portable, as you can see there. The MinION fits in the palm of your hand, connects to your laptop. And so now we're getting some opportunities for doing genomics in the field, which is really good when you want some rapid results. And so this is just some papers that have been done using some of these methods. So they have been done to detect arboviruses within mosquitoes and so forth. It's been used for real-time sequencing of Ebola outbreaks, Zika, and also papillomaviruses in giraffes and so forth. So there is a lot of research starting to come out with these new methodologies. Benefits for the polar regions: they're in field, they're nice, they're small. You can pack them in your suitcase. Literally, people are now building labs in a suitcase. It provides you with the opportunity of real-time sequencing, so they can give you quick and rapid identification. So with some of the work done with the Ebola and so forth, they had results back within sort of about 6 to 8 hours of extracting the DNA and running the sample. They can be used in field laboratories. The devices are a lot cheaper than those bigger machines. They can enable a rapid response if there is an infectious disease outbreak. And they do come with some easy to use platforms for that basic identification purpose. You still need to do some bioinformatics if you want to do more in-depth analysis, but they do have platforms that are easy to use if you just want to know what's happening. There are some limitations, and probably one of the biggest ones is testing and validation is quite limited for wildlife diseases, and also doing that field analysis. There's lots of different kits to choose from, but limited protocols in terms of what is the best one for your scenario. You still need DNA and RNA extractions, so that's still something that you need, but there's a lot of development in that area. So there's a lot more places that are developing kits that you can do in the field. There's currently a project looking at cassava virus in Africa, where they literally use some of these methods to sequence plant viruses on the farm under a tree. So, you know, there are ways, but there is a lot of development still needed in this area. Storage and transport of the flow cells and reagents is also can be a limitation. So having the facilities to store some of these things. And so currently at the moment, a number of the members of the working group and myself are actually starting to do a lot of testing and validation of these new tools for specifically polar regions and specifically for disease surveillance. And we're actually, as part of that testing and validation process, we're also developing our protocols so that, you know, the rest of the Antarctic community and even other polar regions can utilize and start using these processes and start actually increasing our disease surveillance capabilities within the polar regions. So thank you for that, and I think we will go to questions if we have any.
Hi Megan, there is a question. Question is, pathogen discovery, surveillance and monitoring is an important part of a biosecurity framework.
Yep.
Without a biosecurity framework, it's hard to deploy the findings or prioritise and drive the work. Is there any interest in a more structured approach to biosecurity in the polar regions? And if so, how might this be progressed?
That is a
challenging one for you.
It is a challenging one, but it is a fantastic question that we had. So basically recently, so yesterday we had a meeting of the working group, and this is definitely one of the areas that we are focusing on and trying to look at ways that the group can work to help sort of build better biosecurity frameworks and to work with different national Antarctic programs and other stakeholders to improve biosecurity measures down south, but also other ways that we can work together to do this. Beautiful. If people have any other questions, please feel free to put them into the chat box. We do have a few minutes left before. So Andreas, just for the last final couple of minutes, did you want to give a bit of a brief of some of the future directions of the working group and how we plan to start to, I guess, coordinate some of this research in Antarctica? Because I think that's probably one of the most important things at the moment for disease surveillance, as we've pointed out, that, you know, there's not a lot of coordination in this space. It's very sporadic, and that's one thing that the working group is hoping to address and start to facilitate more research.
Okay, thanks, Megan. Well, I think it's been a very good workshop. I think there have been many interesting talks showing how is the situation about the polar wildlife health in Antarctica specifically. And from our working group, our idea when we created this working group was to promote the knowledge of the wildlife health, including parasites, pathogens, and also all the ecological and evolutionary aspect that is related with this topic. And what I can see from these talks is that the information is very few, is very little. There is many different gaps in knowledge. There are very few species where there is some kind of knowledge, but there are still many more species for those we don't know nothing, more or less. There is also some problems with the geographical cover of the information that we have. There is some places that are more or less well studied, but there are many others that are completely unknown. And so this is one of the goals of the group is just to do these kind of things, to communicate how is the level of knowledge that we have of this important thing. And this is important in different terms. It's important in terms of as we have seen in different talks, Antarctic vertebrates, marine mammals and seabirds are sentinels of the marine Antarctic marine ecosystem, and therefore all the knowledge that we can have for these organisms is extremely necessary, crucial for knowing which is the health status of the ecosystem. And disease, as we have also seen in different talks, is one of the aspect that influence very much in the dynamic population of these organisms. So we have to know what happens with the disease, with the parasites, because they influence in the population dynamics, and we know this because we in terms of conservation, because we have to know if there is some microorganisms, for instance, introduced in the Antarctic that is no before, but now the information is very, very, very scarce. So we don't know if we discover a new microbes, we don't know exactly if this is new or there is also present for a long time ago. And the information is also important to discriminate what is the effects of the pathogens and parasites in relation with other factors like climate change, global change, and of course human impact they are naturally included in the functioning of the ecosystem. So our goal is, as I said, to promote this knowledge, to compile of the knowledge, and to encourage researchers working on wildlife to include this stuff, this kind of issues and topics in their research routine. I know that it is very difficult, that this is not, someone is not a specialist in this topic. It's difficult to carry the work by themselves, but the other goal of this group is to coordinate this kind of research. So if some of you are interested in including this kind of research in your projects, in your research goals, please contact us, and we will have now a very good community with different specialists in different species, in different pathogens, parasites, groups, and techniques that I think that we can do a very good collaboration to increase the knowledge. And this is one of the other mission that the working group has from the very beginning. And the other is to try to coordinate and organize the question about protocols. We have to to standardize the knowledge and to have protocols that can be used for different people in different places and for different things. And this is another of the things that how we also have seen in the talks, that there are different approaches, and sometimes it's difficult to put all the data together. And this is another of the mission of the working group. And finally, I think it's the big task of the group is to organize or coordinate or at least encourage all the stakeholders that there is needed surveillance program for knowing what is happening with health and disease in Antarctic wildlife, in order to avoid or to be ready in case there is mass mortality that can be because one novel microbe arrived to Antarctica and can cause mass mortality, and we have to be ready for responding to this emergency. So we need to have good protocols for surveillance, for collecting samples, and to have a good network for afterwards to analyze the samples and to get the correct information about what happened or what has been happening. And this is very important for the preservation and the conservation of Antarctica and the wildlife. And actually is included in the revision of the research goals for the next decades that was done by the SCAR and is also being commented in one of the talks. And this aspect has been included in this relevant question that has to be solved in the next decades. So we, our working group, we are very happy to collaborate with researchers to increase the knowledge of about disease and health of Antarctic wildlife, and with all the stakeholders, the Committee for Environmental Protection of the Antarctic Treaty, COMNAP, IATO, to help in anything that has to be related with these issues. And I think this all the things we want to do, and we hopefully we will get in the next future.
Perfect. Thank you very much. So that comes to the end of our webinar. We first, most importantly, want to acknowledge the support of SCAR and, of course, the expert group of birds and marine mammals, which is where this working group is part of. So we are part of the SCAR community, and so we want to acknowledge their support of the work that we do. And also acknowledge Black Box, who have provided us with this platform today to be able to deliver this webinar to you all. So on behalf of the working group, I want to thank you all for attending. I hope our sessions have been very, very informative, and you've learned a lot. I'm sure there's probably lots of questions and new ideas and stuff that people might have. And as Andreas said, you know, we are always looking at opportunities for collaborations and so forth. So please definitely reach out to the community if you are interested in any sort of disease, wildlife research, or even from our stakeholders if you have questions or things that you would like some advice or guidance. We have a network of lots of different experts within our group that we can connect you with. We have recorded this session, so we will be making this available through the SCAR YouTube channel and webpage. So if you missed any parts of this, or you want to rewatch it, or if you want to share it with anyone, I will send you the link once it is available. So thank you all very much for attending, and hopefully we will see you all soon, and stay safe. Thank you.
So thank you for everyone.
Thank you very much
for organizing.
Yep, thank you. All great talks.
Thank you. Bye-bye.
Yeah. Great talks. Yeah.
Thank you. Take care.